Hello students. Today we are going to discuss about very important structure or a cell organelle, and that is cell wall. Cell wall is a non-living rigid structure. Okay, and it forms an outer covering from the plasma membrane of a bacteria, fungi, alga and the plants. So each organism, whether it is a bacteria, fungi, alga or a plant, they will have a cell wall. Okay, only cell wall is absent in animal cells. Then dinoflagellates, you can see here, this is the dinoflagellate, and that dinoflagellate cell wall has a stiff cellulose plate on the outer surface. This is the characteristic of the cell wall of dinoflagellates. Whereas in case of euglenoids, here you can see the euglenoids. Euglenoid, instead of the cell wall, in place of a cell wall, they will have a protein-rich layer, and that is called pellicle. Okay, that is a cell wall like a structure, and that is a pellicle which makes their body flexible. Here in this slide you can see a types of a cell wall. So cell wall can be divided into three groups: primary cell wall, secondary cell wall and tertiary cell wall. And further you can see here cell wall, which is secondary cell wall, further can be divided into three groups: S1, S2 and S3. So now let me discuss one by one. What is a primary cell wall first? So what is a primary cell wall? Primary cell wall is a thin, elastic and main component of a primary cell wall will be cellulose. Along with the cellulose you will see a hemicellulose and the pectin. Okay, and it will diminishes as cell is maturing. Then it is capable of growth. So primary wall is capable of growing. And now let me discuss about cell wall, which is a secondary cell wall. It is rigid, thick. Okay, as compared to primary, it is rigid and the thick. Then it is composed of cellulose, hemicellulose and the pectin. Composition will be same as per the primary cell wall. Then it will absent in meristem cells. Okay, then inner side of a primary wall towards the cell membrane, it will be inner side of the primary wall. We will discuss in next slides. Okay, so it will be at the inner side of a primary wall towards the cell membrane. Then what is a tertiary cell wall? Tertiary cell wall will only present in tracheodes of gymnosperms. Okay, gymnosperm is a group of plants. Okay, we will discuss in chapter number 3. Okay, so gymnosperms is a one group of a plant in which only you can see a tertiary cell wall, and it is composed of hemicellulose and the xylem. This is the main component of the tertiary cell wall. Now we can also discuss about middle lamella. So what is a middle lamella? Middle lamella is a common layer between two cells. Suppose one cell and second cell, in between these two cells one layer will be there, and that is called middle lamella. Okay, it is in between two cells, and that's why name is given middle lamella. Middle lamella is consist of calcium and magnesium pectate. Okay, how middle lamella form? What is the function? What is the different type of organelles involved in the formation of middle lamella? We will discuss in chapter number 10 during cytokinesis of a plant cells. Okay, so middle lamella is consist of calcium and magnesium pectate and plant cement. It will be act as a plant cement, and amount of calcium is more. So here in the structure of a middle lamella, calcium proportion is very high. Now let me discuss more by using this diagram, which what is the cell wall in its type. So here you can see this is the primary cell wall, and this is the cell wall structure. Here you can see this layer is the primary cell wall. Okay, this is cell number 1, this is cell number 2. Here you can see in between these two cells, you can see here. Okay, that is a middle lamella. Okay, that is made up of calcium pectate as we have discussed in previous slide. Okay, then you can see here a tertiary cell wall structure. Okay, in between that a secondary cell wall is there. So by this way primary cell wall is constructed, then secondary cell wall and the tertiary cell wall. Then you can see plasma lemma, or we can say cell membrane is found. Okay, so this is the middle lamella, primary cell wall, secondary cell wall and tertiary cell wall, and in innermost layer will be plasma lemma. Okay, so by this way, all cell wall will be arranged on the cell. Now let me discuss about different type of cell wall present in a different kind of organism. So first of all, in the alga have cell wall made up of cellulose, galactans, mannans and the minerals like calcium carbonate. So different type of organism will have different composition in their cell wall. So alga will have specifically a cell wall which is made up of cellulose, galactans, mannans and the minerals like a calcium carbonate. While in case of plant cell wall, composition will be different. So in case of a plant cell wall will be composed of cellulose, hemicellulose, pectins and the proteins. Okay, and cell wall here worked as a frame or a protective layer of a cell in case of a plant cell. Cellulose microfibril and microfibrils, okay, microfibrils and macrofibrils are arranged in the layer to form skeletal of the cell wall. Okay, two type of fibril together is includes microfibrils and the macrofibrils together arranged in the layers, and finally it will form a skeleton of the cell wall. In between these layers, one substance means other substances like pectin, hemicellulose will be arranged or may be present. These form matrix of the cell wall. So by this way, cell wall materials like a hemicellulose, pectin, lignin, these all important component will be formed or synthesized by using Golgi bodies. This is one type of a cell organelle. Okay, in the case of a plant cell, it is considered as a dictyosome. So we will learn in this chapter in next lectures. Okay, what is the Golgi body? What is the structure of a Golgi body? What is their function? Okay, in detail in next lectures. Okay, so here let me repeat. Cell wall materials like hemicellulose, pectin, lignins, all these components will be synthesized in the plant by using Golgi bodies, or we can say dictyosomes. Formation of a cellulose occur in the cell membrane. Okay, this cellulose formation will occur inside the cell membrane, whereas rest other things will be produced by the Golgi bodies. Materials of the lipid nature means whatever are the lipid in content. Okay, lipid in nature like cutin, suberin, that is also a part of a cell wall. Okay, cutin and suberin, that is a wax-like material. So if nature of a material is a lipid type or lipid in nature, so such a type of a component will be synthesized in the spherosomes, a special structure which is responsible for formation of a lipid in nature component, especially cutin. Formation of a cell wall occur by two methods. Out of that, first is the intussusception and second is the appositions. So let me see what is the intussusception. Intussusception is the method in which a deposition of a cell wall material in the form of a fine grains occur. Okay, so fine grain will be arranged like that and layer will be formed, and such a type of a method is called intussusception. Then second is the apposition, and it is the layers deposition, means it will be form a layers. Okay, not in the form of a fine grain, that will be form a layers and it is called apposition. So primary wall is a form mainly by intussusception, means by granule. Okay, and while secondary wall formed by both the method, whether it is means it will include both the method, intussusception and apposition both. Growth of already formed cell wall occur by only intussusception. Okay, so by this way you can see which type of method will be applicable. A special protein called expansion, okay, help in the growth of a cell wall by losing the cellular microfibril, and addition of a new cell wall material take place in the space. Okay, very special type of protein expansion helping in the cell wall expansion or a growth. Okay, thus expansion is a called cell wall loosening factor. Okay, because here you can see it will losing the cellulose microfibrils and addition of a new cell wall material take place. Okay, it will losing the microfibrils and it will add some new material, and that's why expansion is called cell wall loosening factor. So here in this diagram you can see outermost layer. Okay, that outermost layer you can see here. Okay, and that is the middle lamella, as we have discussed. That is means common in between two cells. Okay, so this is the middle lamella. Inner than the middle lamella, inner side of the middle lamella, there will be primary cell wall. Then you can see three greenish layer. Okay, that is a secondary cell wall layer. That is S1 first, then S2 and S3. Okay, and then innermost side, whatever the space is there, it is called lumen. Okay, so by this way, a plant cell wall formation will. Now let me go through whole process of a cell wall formation by using this diagram. So first of all, here you can see this is the plant cell. Okay, and the microtubules will be arranged in the granules at the both side, and then in the next step, Golgi vesicle, that is also called dictyosome in case of a plant cell. So Golgi vesicle will be arranged in the middle of the cell. Okay, and here you can see spindle fiber that will be helping the Golgi vesicle to arrange in the middle side. Okay, and then this Golgi apparatus will form phragmoplast. Here you can see in the early phase of a cell it will form a phragmoplast, and due to phragmoplast you can see this cell composition will be divided into, or we can say fragmented into, two portions, and that's why it is called phragmoplast. Okay, and in next stage you can see here new cell wall component will be arranged, and here you can see microtubules will be arranged like this. One important structure related to cell wall is a plasmodesmata. So let me see what is the plasmodesmata. The name plasmodesmata first of all proposed by Strasburger. Okay, and in 1901, these are the cytoplasmic connection between two adjacent plant cells. Here you can see these are the two adjacent plant cells. Suppose this is a plant cell A and this is plant cell B, and in between one communication channel is there, and this communication channel or a pathway is called plasmodesmata. Okay, in this plasmodesmata are a characteristic of multicellular plant, and they maintain continuity of cytoplasm in between adjacent cells, and by this way they will communicate with each other. Okay, and they can also exchange some materials with each other. An ER tubule, that is also called desmotubule, will help to maintain continuity of the cytoplasm. Now let me go through some important functions of a cell wall. So first function is to give shape to the cell. So cell wall is responsible for giving shape to the cell. This is very important function. Then second is it is protecting the cell from mechanical damage or infection from bacteria, viruses and all that. So cell wall is importantly play very important role for the protecting the cell from the infection. It also help in the cell-to-cell interaction, as we have discussed in previous slide. A plasmodesmata structure play very important role for the interaction between adjacent cells. It provides barrier to undesirable macromolecule. It also provides some barrier to undesirable macromolecule to come in or come outside. Then let me discuss about the differences between primary cell wall and secondary cell wall. So first difference, in the primary cell wall you can say it is made up of cellulose microfibril and it will arrange in a dispersed manner. Okay, whereas in this case, in case of a secondary cell wall, microfibrils are parallel to long axis of the cell. This is the main difference. Now in the primary cell wall, hemicellulose more, or its amount will be high, that is a 50%, and here in secondary cell wall you will see hemicellulose will be present but in the lesser amount. It will be only 25%. Primary cell wall have a lipids 5 to 10% and the protein will be 5%, whereas in this case proteins and lipids either absent, may be absent, or in the little amount than the primary cell wall. Second thing, it will form, means the primary cell wall will be formed by intersusception method, whereas the secondary cell wall will be formed with both the method. It is intersusception also and the apposition also. Second and last point, that is a primary wall is a universal layer. It will found in each and every cell, whereas in this case, means in secondary cell wall is absent in the meristem cells. This is the main differences between primary cell wall and secondary cell. This may be asked in the board examination in 2 to 3 marks. This is your homework for today's lecture. Just you have to write down in your notebook and sub. And before we finish the lecture, let me summarize all topics by using one animation video. So look at this video.
Our Earth is home to diverse species of plants and animals. Yet, they all have a common structural unit. The eukaryotic cell, or a cell which possesses an organized nucleus surrounded by a nuclear envelope. This nucleus, containing chromosomes that carry the genetic code of the organism, is surrounded by cytoplasm, a semi-fluid matrix which makes up the volume of the cell. Present in the cytoplasm is the cytoskeleton, an elaborate network of filamentous, proteinaceous structures that gives the cell its characteristic shape, aids in intracellular transport, gives mechanical strength to the cell, and also helps in motility in case of motile cells. The cytoplasm also contains numerous single membrane-bound minute vesicles called microbodies, which are filled with enzymes that aid in the synthesis of amino acids. Also embedded in the cytoplasm are several organelles or membrane-bound structures which lead to its extensive compartmentalization. However, the cytoplasm of all eukaryotic cells does not contain the same set of organelles. Plant cells, for instance, have a large central vacuole and plastids, whereas centrosomes are only found in animal cells. The centrosome, surrounded by amorphous pericentriolar material, is made up of two cylindrical structures called centrioles that lie perpendicular to each other. Each centriole is made up of 9 evenly spaced peripheral fibrils of tubulin protein, with each peripheral fibril being a triplet. All adjacent triplets are linked to each other, forming a cartwheel-like structure. The central part of the centriole, called the hub, is proteinaceous and is linked to the tubules of the peripheral triplets by radial spokes made of protein. Centrioles form the basal body of cilia or flagella and spindle fibers that give rise to a spindle-like apparatus during cell division. Eukaryotic cells differ not only in their cytoplasmic organelles, but also in their structure. Take, for instance, animal cells in which the plasma membrane forms the outer boundary of the cell. But in fungi and plant cells, the plasma membrane is covered by a cell wall which forms the outer boundary. Interestingly, the composition of the cell wall is different in different eukaryotic cells. In algae, the cell wall is made up of cellulose, mannans, galactans, and calcium carbonate, whereas in fungi, it is made up of chitin and glucans. On the other hand, a plant cell wall is made up of cellulose, hemicellulose, pectins, and proteins, consists of 3 layers: the middle lamella, primary wall, and secondary wall. The middle lamella, mainly made of calcium pectate, lies between neighboring cells and glues the cells together. The primary wall is the first formed wall that develops when the cell is growing. It is thin, elastic, and extensible. However, its growth capability diminishes as the cell matures. The secondary cell wall develops after the cell has matured. It is formed on the inner side of the cell and faces the plasma membrane. Interestingly, the secondary cell wall and lamellae do not occur as continuous layers, as they are interrupted by plasmadesmata, fine strands of cytoplasm that facilitate the transportation of materials between different cells in a plant. The cell wall is an important part of the cell as it lends shape to the cell, protects it from mechanical damage and infection, prevents the entry of undesirable macromolecules such as nucleic acids and also aids in cell-to-cell interaction. Though a cell wall may be present or absent in different types of eukaryotic cells, these cells are the basic unit of life in many organisms, including plants and animals.
In next lecture, we will learn important structure or a cell organelle, and that is endomembrane system, that is also called ER system. So till that, goodbye.